Okay, guys. So the first thing I'm going to ask you guys to do actually is on that paper. In the little space in the upper right-hand corner where there's a little bit of margin, I'm going to have you guys draw a model, a very simple model. Do you guys remember what the main component is of the membrane, plasma membrane? What's the main component that's there? What is the membrane made out of? It's made out of a couple of things. I'm looking for the main component. I know this is going back to Monday. It's not fresh in your brains. Tuesday's kids were whipping it out because they had just seen it Monday. Wednesday it was like some remembered, some didn't, and like nobody's remembering today, which is okay. But you don't remember what that main structure is that's found in the membrane? All right, so let me help you out a little bit. If I look at this, this is a model of this plasma membrane. What's the main thing or the most abundant thing that you see there? Not the biggest, but the most abundant. What do you think? Cholesterol. Okay, so there is a lot of cholesterol there, but I would argue something else. The phospholipid, right? I see a bunch of these guys. Okay, so what I'm going to ask you guys to do is draw that simple model of a phospholipid on the top right-hand margin of your paper. And it's a phospholipid. The phospho part of that is due to the phosphate group. And where do we find the phosphate group? In which part?
The surface part.
Okay, so we got the phosphate group up here. The lipid part or the fatty part are going to be these tails, right? That are the fatty acid tails. They kind of do more of that hydrocarbon chain thing, like that. Be more accurate. And one of them might be bent because it's unsaturated. All right, and then the next thing I want you to do is label the hydrophobic and the hydrophilic regions. So can somebody tell me which part is hydrophobic and which part's hydrophilic?
The top part of the phosphate is hydrophilic, and the lipid part is hydrophilic. I mean hydrophobic.
Hydrophobic. Okay, so it's hydrophilic because it's got that charge, right? It's polar, and it likes water because water is also polar, right? So it's hydrophilic because it's polar. The hydrophobic region doesn't like water because it's non-polar. We have this saying, and it'll come up later in the discussion. It's called like dissolves like. What that means is polar things will mix well with each other. Non-polar things will mix well with each other. But polar to non-polar do not. And that's why you get that oil and water scenario, right? The oil droplets that ball up in water. And then one other term I kind of wanted to point out to you. It showed up in the Khan Academy assignment, but if you weren't really looking for it, you may have kind of just glossed over it. But this molecule is considered amphipathic. I think that's the right way to say it. But it's amphi because just like an amphibian can live in the land in the water or on land, this guy kind of has that dual nature to it where parts of it are polar and the other part is non-polar. So we've got this amphipathic nature or dual nature. Okay. So if I were to ask you this question, where did it go? There we go. If I were to ask you question number 1 on an assessment, which of the following diagrams shows the correct arrangement in a phospholipid bilayer? What are we going to say? Which one do you think it is? Yeah. Okay, so we just thought a second ago, right? A, we can get rid of right away because there's only one, right? We need 2 for the bilayer, so that one's not going to work. So let's talk about why it's B and not C.
Because they connect by the nonpolar fatty acids.
So these guys are going to be attracted to each other, right? And what do we know is it possible, like, these guys would be attracted to each other too, right? But we don't see this happen because what do we know is what are we gonna find on both sides of the cell membrane, both inside and outside the cell? There's a lot of water, okay? So what's happening is these guys are trying to stay away from water, right? And they're finding each other, and these guys are attracted to water, so they're more easily facing that direction.
Well, I was gonna say technically C also can be the right choice if there was another layer on each of the sides facing each other.
Oh, so then we would have like a double membrane thing. Yeah, yep, do that. All right, so that one's gonna be B because of that like dissolves like scenario. Okay, so now that we've talked about some of the overall structure of the phospholipid bilayer or membranes, we're going to look at how the structure allows it to do its function, right? So there's that theme again of how the structure determines the function or allows the function. Do you guys remember what some of the functions are of a cell membrane or plasma membrane? If I were to quiz you out on organelles, the cell membrane always shows up as one of the organelles, right? What is its role? What is its job?
Keeps it like together.
So it creates a boundary, right? That outer boundary that sort of separates inside the cell from outside of the cell. Good. What else? So let's say I have my boundary. I have inside and outside of the cell.
It protects the cell.
It protects it.
Helps determine what goes in and out.
Perfect, right? It can control what comes in and what goes out. So that's a key thing. All right, so that's where we pick up on your paper. We see that membrane function, how does the cell membrane control the movement of materials? So why? So big picture, what's the importance of this? We know that the membrane is critical to the maintenance of homeostasis in living organisms. Now, I want to take a second and just talk about what that means. Homeostasis. Have you guys heard that term before? If you're in health careers, you would have. Balance. An internal balance, right? So despite changes going around in your outside environment, we maintain a stability or an internal balance. Just to give you a couple of examples of what homeostasis is and why it's really important. Blood sugar levels, right? How much glucose you have in your blood. Our bodies like to maintain a certain narrow level of blood glucose levels. If your sugar levels get too high or they get too low, your what goes crazy?
Your other vitals change.
Your vitals change, right? So it can be harmful to your body if it skews too far, too high or too low. Does anyone know what that's called when people's bodies don't have the ability to self-regulate the glucose levels? Diabetes, right? If you are not able to maintain homeostasis of blood sugar levels, you're a diabetic. And again, that can have serious consequences, like you said, creates all sorts of problems. Body temperature, another good example of how homeostasis is a key role, right? Our bodies like to work and operate at an ideal temperature really close to 98.6. What happens if I go out of that range too far? If I get too hot and I start approaching 102, 103, 104, what could eventually happen to you?
You could pass out or you have a fever or seizure.
Fever, seizure, eventually, if you can't control it, thankfully we have medicine that now lets us control it, but yeah, you could die from it, right? And again, if your body temperature gets too low, you can die that way as well, right? So that's where we're going with homeostasis. Somehow the membrane is allowing us to maintain that balance at the cellular level, okay? So the cell membrane separates the cell from the external environment, like we mentioned, and plays a critical role in regulating the movement of materials into and out of the cell. So that's talking about its function. Additionally, eukaryotic cells are made complex by the presence of internal membranes, okay, that form the organelles. Remember, an organelle, the definition of it is membrane-bound structures that perform a specific job inside the cell, right? So we know we've got internal membranes that form the organelles so that cells may become specialized. These organelle membranes create the compartments within the cell that can do specific functions. So we're talking about the whole cell membrane and also the internal membranes. Hopefully everybody was able to read those two sentences and know what we're talking about, right? A lot of this came from our previous unit where we talked about compartmentalization, right? And we talked about organelles that are found in eukaryotic cells that allow them to be specialized. For example, muscle cells have the abundance of mitochondria, okay? So good. Hopefully that was easy to understand. So, before we talk about how it controls what goes into and out of the cell, we need to look at what type of molecules are going to have to come and go, okay? What are we trying to get into and out of the cell? So that's where model 1 comes into play. So we've got 4 different types of molecules, okay? So number 1, consider the ions and molecules above. Identify at least 2 substances up here that would need to move into the cell to maintain homeostasis. So what things do our bodies need to take in, or our cells need to take in from that chart? Oxygen is going to be 1. What else do we take in? We like to.
I was gonna say sugar.
Yeah, right? So the 2 big things that we have to do to stay alive is to breathe and eat, right? So glucose. And you could also argue water, right? We need water. So why do we need each of these substances? Oxygen and glucose, what's the main process that those guys are involved with? They are, actually. So you're good, but let's, photosynthesis is reserved just to plant cells, right? So all cells need to do another process.
Respiration?
Cellular respiration. Basically, right, we have nutrients. We need to take in nutrients. In this case, they're associated with cellular respiration. Now let's talk about the ions and molecules that would need to move out of the cell in order to maintain homeostasis. What are we going to get rid of? What are we going to expel?
Carbon dioxide.
Okay, so carbon dioxide is 1. I'll give you the other one, unless, or you want to take a stab at it? No, it's all right. I'll give it to you because it's not an obvious one, but urea is another one that we have to get rid of. Do you know where urea is found? Urine, right? Good. So it's being expelled through urine. That's actually what gives pee its color and smell. And it comes out through the urethra. It's a little tube, right, where it comes out. So why do you suppose we're getting rid of these substances? What could we consider them? Waste products, right? They're essentially waste products from different metabolic processes. We talked about that a little bit when we did the cell size activity, right? We talked about how it's beneficial for cells to be small because they're more efficient at getting those nutrients in to the cell and getting those waste products out. Okay? And so the last thing that they're asking us to do on that first page is to complete the table, these last 2 rows in the table, and identify whether we're looking at polar, nonpolar molecules, or if we consider them large or small. So for type 1 ions, I know we haven't really gotten into a lot of this yet, but the fact that I see pluses and minuses kind of tells me that they're polar, right? And they're actually very polar, highly charged. And they're small because they're at the atomic level, right? Just the size of an atom. They tell you that glucose is polar and large. I'm going to put a little star next to this guy because he's very similar to the phospholipid, where there's regions of both, right? There's some polarness to it, but there's other areas where it's more non-polar. So it's got a little bit of both going on. How about type 3? If water's present, then I know type 3 is going to be polar. Good. Would you consider them large or small? Compared to glucose, they're small. And to all the other organic macromolecules that we looked at, they're pretty small, right? These guys are going to be nonpolar and small. Okay, so we have 4 different types of molecules, all with different sort of combinations of characteristics. Okay?
What did we put for explain the source of the molecule?
For this one,
Yeah.
2B, it's just because they're waste. They're a waste product. Okay, so we know which molecules, we know some different categories of molecules, right? Some different types of molecules, and we know that we need to get the nutrient ones in and the waste products out. So now we're going to look at how the cell is selectively permeable, right? And controls what goes in and out. So I just touched on it very briefly on Monday's Zoom lesson, but does anyone remember or have some idea what selectively permeable might mean?
There's certain things that go in and out of the cell.
Yes, and other things that can't, right? So it kind of determines what can pass and what can't. So the example I always like to give is the kitchen strainer. When you boil water and I dump it out into the strainer, what happens? Water goes through, pasta doesn't, right? So that's a selectively permeable, or sometimes it's called semi-permeable barrier. Window screen, the same thing, right? The openings allow water to get in and out, not water, air to get in and out, and water if you're spraying a hose. But it keeps other things out like the bugs and bees and everything, and it keeps you from hopefully coming out the window too, right? All right, so we have a selectively permeable cell membrane. So in number 4, it's telling you that the 4 diagrams in model 2 illustrate movement of the 4 types of substances that we saw in model 1 across the phospholipid bilayer. Use your knowledge of the membrane structure and chemical structures in model 1 to identify the shapes that are used in model 2. So now we have to identify or really analyze what it is that we're looking at in these models. So right off the bat, I know this one's going to be phospholipids. So you might have to flip back to the first table to see if you can identify what these are trying to represent based on their structure. I feel like the easiest one is this hexagon structure here. What would we say that one is?
The glucose.
Glucose, right? It's got that ring structure to it. Can you figure out any of the other ones?
The circle with the cross in it, I said ions.
Ions, yep. So notice that up here it's got pluses and the minuses to represent both charges. All right, did you have one?
I was gonna say that one.
Oh, you're gonna do the ions? Okay. How about this spokey one? I got an up and 2 sides.
Cholesterol?
Well, so, but it's cholesterol. Did you say cholesterol? Yeah, so the only thing is cholesterol is not one of the molecules that we saw in table 1. So pick from that. Close. If it was, we're getting there. If it was water, it would just be that. So what's the other one?
Carbon dioxide.
So we're gonna put carbon dioxide over here. So it's got the carbon in the middle, the 2 oxygen on the side.
Oh, the urine one.
That's the urea, right? The SCOBY one is the urea one. If you look on your paper, you notice how the there's a carbon in the middle, oxygen goes up, 2 amine groups to the side. Okay. All right, so we can see, we can identify all the different things going on inside of the diagrams. Okay, and then the next thing you're being asked to do is to label the diagram, or each diagram, with the ion type or molecule type. Okay, so now that we know these are ions, this is going to be type 1, right? Very polar, small molecules. Over here, if this is glucose, we consider that type 2. which was large and polar. Urea fell into type 3, which was small and polar. And then this one's going to be type 4. Carbon dioxide was nonpolar and small. Okay? So we're sort of diagramming each one of those 4 scenarios. Then the last thing we need to do is identify on the diagram, it wants you to circle the side of the membrane where the ion or molecule would have originated in the normal function of a cell. So that goes back to that first 2 questions. We're asking, which ones are trying to get in, which ones are trying to get out. So I'm gonna skip type 1 because we haven't talked a whole lot about ions yet. Don't have a whole lot of background knowledge on that. Let's look at type 2, which is glucose. Is it trying to get into the cell or out of the cell? In. So it's going to start on the outside trying to get in, right? So is inside or outside on the left or right on here? Or if I'm in the extracellular fluid, where am I? Inside or outside? Outside. So if it's trying to get in, it's going to start on the outside and try to move in, right? So that's what we want to do. It's here, it's trying to go that way. For the urea and the carbon dioxide, we know that those are both waste products that are the result of some process, metabolic process going on, and they're trying to get out. So they start inside, and they're trying to move their way out. Like I said, the ions I'll give you, they start outside in this scenario, and they're trying to get in. So all we've done so far is sort of analyzed what we're looking at, right? What the diagram is telling us. Numbers 6 and 7, we're going to analyze what's actually going on, like what the diagrams are trying to tell us. So for number 6, it says, assume the substances in model 2 were on only one side of the membrane to start. The diagram illustrates what happens after time has passed. So the diagram is showing us the end result, okay? The end result. Okay? So for 6A, it's asking you which of the molecules in model 2 appear to be completely blocked and we're not able to pass the membrane at all. Ions, right? So they start here, time passed, and this is the end result. They stayed here, right? So they couldn't get across at all. So for 6A, the ions are type 1, did not get across. For 6B, which substances appear to be able to pass freely through the membrane? Which ones seem to move with no trouble whatsoever? What do you think? Yeah, carbon dioxide or type 4, right? And I can see that it's spread out almost equally on both sides. So these guys had no trouble getting across. Good. So, which substances in model 2 appear to pass through the membrane, but with some difficulty? Urea, definitely, right? And we could also argue glucose, right? They make it over a little bit, but not as easily as carbon dioxide. So I would, for C, put both glucose and urea, or type 2 and type 3. Now, for 6D, it says urea appears to pass through the membrane a little bit more easily than glucose does. What characteristic of urea might help explain this observation? What do you think? It's liquid? Okay, it's liquid, but so let's go back to the table though. I want you to look at the properties that we're talking about on that table. And what's one of the differences between type 2 and type 3, or the glucose and the urea?
Type 3 is polar and
Yep.
4 is non-polar.
For example, okay, so that's a difference between these two that might have a factor in the difference of permeability. One more try. But the difference between these two, these guys are both polar, yet this one moves a little bit easier.
Urea is smaller.
It's smaller, right? So its small size allows it to move just a little bit easier than the larger glucose. Okay, so we can see the result, right? We can start to see the selectiveness of it and the permeability of these different molecules, but we haven't really established as to why that's happening just yet. We talked a little bit about size. So in number 7, notice the little key, right? That's going to be sort of like the key takeaway from this. It tells you that the majority of the membrane is made up of nonpolar hydrocarbon chains. So what's it talking about? This region right here, right? We know this is our nonpolar region of the membrane. Use the diagrams in Model 2 and the table in Model 1 to explain the permeability of the membrane for each of the 4 types of substances in Model 1. Hint, like dissolves like. We talked about that earlier, right? Polar likes polar, nonpolar likes nonpolar, but the two don't mix well together. So how does that help explain what we're seeing with the permeability? So for type 1, what could we say? How come they weren't able to get across at all? This is non-polar region, right? What did we say these guys are? Very polar. So it's like hitting a wall, right? They will not mix with that whatsoever. So I'll give you a second to write that down so we know, again, that the type 1 very polar ions will not mix well with that non-polar region. Okay, does that explain a little bit why we're seeing what's going on with type 2, right? The glucose. We see again, it can get across maybe every once in a while, but for the most part, it is blocked as well. So it's polar and large, so it's being blocked. Why do you suppose every once in a while one can get across? Remember I had you guys star the little polar at the beginning, and I said
it has some of both.
It has some of both. So every once in a while, if it hits it just right, you might get one to pass through. Right? But for the most part, because it's large and has that polar property to it, it's also going to be blocked for the most part. Okay, and then for type 3, we can see that it's a little bit easier. There's still some resistance. The resistance comes from this being polar and not being able to mix with the non-polar. But at the same time, they're small. So again, they can kind of eke through every once in a while. But we're seeing that the polarity, right? The polar, the non-polar, and size is playing a role in all of these. Okay, so for type 3, they're polar, so again, some resistance, but small enough to get through every once in a while. So why do we see type 4 going through with no trouble at all?
It's non-polar.
It's nonpolar, so it can mix well or get through the nonpolar region easily. And it's kind of nice because it's small as well. Right? Makes it go easy too. So I'm going to ask you guys 2 questions, okay? You might see these questions again. The first being, what property or what characteristic of the cell membrane allows it to be selectively permeable? Yes, good, you're on the right track, but more specifically, it's that non-polar region, right? It's the non-polar region that's creating the barrier, okay? And what are the 2 properties of molecules that determine whether they can pass through or not?
Their size and if they're non-polar.
So size and polarity is what determines whether a molecule can get through or not, okay? Very good. Okay. All right, so we're gonna move on to the next model. It's kind of nice actually that things like carbon dioxide and oxygen can just go across very freely because, you know, those are really important molecules to get into and out of the cell, right? And so the fact that they can get across very easily means, you know, we don't really have to do anything to make it happen. It just happens naturally for us. Which is where this little stop, read this thing comes into play. It's going to give us a little bit of information here, okay? So diffusion is the process of molecules traveling through a membrane barrier from locations of high concentration to a location of low concentration, okay? So, and the driving force for this process is simply the natural movement of molecules in random directions. So what do we mean from high to low? I don't know if you've ever seen diffusers that they use in your bathroom, right? Sometimes there's a little machine that's called a diffuser too, and it gives off like a little steam. But if you think of the little glass jars that have oil in it with the 2 sticks that come out and they call them diffusers, what happens? The molecules, the scented molecules in there, right, go from the areas of high concentration in the jar and they spread out throughout your room and you get a nice smell that gets carried throughout the room, right? That's diffusion, high to low concentration. I actually set off the fire alarm last year trying to demonstrate diffusion because I sprayed a scent and I usually will say, okay, tell me when you can smell it, and eventually it works its way across the room and the kids, you know, will eventually raise their hand saying, I can smell it now, and we can see diffusion happening. I happened to spray right in front of the light of the projector, and that sensor right there, I thought it was smoke. Sorry, sent us all outside. First time in 12 years.
It was let it cross.
Okay, so but notice that even with the diffuser, you don't have to turn anything on. That happens naturally because the driving force is simply the natural movement of molecules in random directions. So what does that mean? Well, you know, all matter has energy associated with it, right? The atoms in this solid disk are actually in motion, right? What are they doing? They're just kind of vibrating next to each other. When you get to liquids and gases, they start moving a little bit more with liquid. They're kind of rolling around each other. And then in gases, what are they doing? They're all over the place. They're separated and they're spread out. But there's no, it's all random, right? There's no set motion for these things. They're just kind of naturally have energy associated with them and they're moving around. So that's what's happening during diffusion. They're naturally moving around and things like to spread out, right? Go from areas of high to low. So whether the molecules are allowed to cross or not is only due to the polarity of the molecules and their size. We just talked about that. And no energy is needed. It's due to that constant random motion. No extra energy is needed, which is why diffusion is considered a passive transport. So what we need to know out of this is that diffusion means molecules are going from a location of high to low concentration, and that no energy is needed. It's due to that random motion that's already there, and it's considered a passive transport because of that. So to kind of summarize that, passive transport means no energy is required, and diffusion is an example of that. Okay? Perfect. Now, Model 3 is talking about embedded proteins. Remember that proteins are another component of the membranes, okay? So we have embedded proteins. So what on these three diagrams is trying to represent the proteins? What do you think? Yeah, the two gray bars, right? So that's what number 8's asking you to do. We want to label the embedded proteins. So we can see that we go this one on that diagram, this one here. And of course, we've got the one in the top right over there as well. So compare what you see. Let's say this is the end result again, right? We're looking at what's happened over time. What appears to be the effect of inserting a protein channel into the membrane on the movement of molecules?
It creates, I don't know how to word it. I was going to say it creates a space so it allows them to slip in.
Perfect, right? It's allowing molecules to move across easier. So things that were not able to move at all, or things that had difficulty moving can now get across easier. And I'm going to okay. So I've highlighted different regions of the protein up here, okay? So is the inner surface of the embedded protein, where you see the red, is the inner surface of the embedded protein likely to be polar or non-polar in the examples? Polar, right? And why? Let's explain that. Why would we say that it's polar? It goes back to that like dissolves like scenario, right? So because this is polar, those polar things can now just easily get across. Almost creates that pore, that opening. I had an excellent question from a student in the last class asking, does this prevent those non-polar substances from getting through? So that's it, like he's analyzing things, so that was good. What I mentioned to him was the fact that those non-polar things are now not going to be able to pass through this little region, but guess what? They're still going through on all the other parts. Okay, so they can still get through, not through the embedded protein part. So this guy's just helping out the polar molecules. And I'm going to take a second and tie this into our previous unit, okay? And if the bell rings on me, ladies, do me a favor and just make sure you read this section. You don't have to answer questions 11 and 12, but I want you to kind of get a head start on what we're going to cover next. So make sure you read this part. But for this, I want to tie it into our biochemistry unit, right? So we've got proteins here, and those proteins are almost that amphithetic nature as well, where there's some polar regions, some non-polar regions. So do you guys remember what the building block of proteins are? What's the monomer? Amino acids, right? And we know all amino acids have that same general structure. We've got the central carbon, an amine group, carboxyl group, right? So if these are essentially made up of a chain of amino acids, what is it about the amino acids that allows the protein to have both polar and non-polar regions? Here's your amino acid. what on here is going to determine, because remember, it's a bunch of these chained together and then folded in on themselves.
The R group.
The R group, right? It's the properties of the R group that's going to determine, because this can change depending on what R group is there. It's going to say, okay, so the ones here are going to be nonpolar. They have to be because they're in that nonpolar region of the membrane, where the ones here are going to be more polar, okay? So that kind of ties into what we talked about in that first unit. And so read this section, since the bell hasn't rung on me yet. I just want to point out that it's summarizing a little bit of what we just talked about. What you're seeing there in those images is something called facilitated diffusion, okay? So when an embedded protein assists in the passive transport of molecules in the direction of the concentration gradient, so again, going from high to low, it's called facilitated diffusion. So it's still no energy required, it's passive. It's still going from concentration of high to low, but because we have little helpers that are facilitating the process, and what are those helpers called? We've got the embedded proteins that are giving us the assist, right? It's now called facilitated diffusion. Still passive. It goes on to talk a little bit about active transport. We'll get into that in a whole other lesson, so you guys are good to go.